I'm Meredith Blackwell, and I'm here at the International Mycological Congress 12 in Maastricht, Netherlands. And I'm going to interview Dan Henk, who is someone I've known since he was 6 years old, maybe. And we're going to talk about his career in mycology, and he has a little bit different trajectory from most Americans, but we'll get into that. So, Dan, would you start out telling us where were you born?
Where was I born? I was born in Athens, Georgia, where both my parents were students at the University of Georgia, and that's where I was born.
Okay, now you had a really early contact with mycology, but I don't know if you knew it at the time, but through your mother?
Yeah. So the story my mom tells me, I don't know, but the story she tells me is that when I was very small, she was studying. So the University of Georgia had amazing mycologists at the time and had a whole program, and part of what they did was capturing aquatic fungi. And what my mom did with my little baby hares was use them for baits. So she would actually, she was part of the team working in the mycology labs at the time, and so apparently my hair ends up in some micrographs somewhere as a small child. So maybe it was destiny. I didn't think so. But
that's an interesting twist. And your mother actually was working with Mel Fuller,
That's
who
right, yeah.
worked with those spark fungi.
Exactly.
And they swim toward cues. And I guess your baby hair was a cue.
Exactly. So I think it's the baby hair has the right sort of like lipid composition that works to get the spores. But I never really learned very much about it. I just heard the story that my
And your father was a biologist, but not working with fungi.
No, at the time he was studying some, I guess they would have been called protists at the time, but they were both very focused on the technology at the time as well, which was electron microscopy, and so they were doing a lot of that. And that was also my mom's work with Mel Fuller was also around the imaging and microscopy.
Yeah. So you're one of the few mycologists I know that has parents that were biologists and a mother who studied fungi even. So,
yeah. I don't know if that's unusual. I don't
it
know
is,
as many mycologists as you, but yeah.
I think it seems to be at least a little bit older mycologists never had parents that knew what mycology was usually. So when did you get interested in fungi? Was it much later?
No, that's all you, Meredith. So I went to university. So we moved to Louisiana, and I started university thinking I was going to do horticulture, basically. I would love flowering plants. I tell a lie. So I was interested in fungi, again, kind of like before I knew it. So actually, Professor Tucker took me on an excursion with my mom, who was in the class at the time, and I loved lichens. It took me a minute to remember that, but I did like my first grade science project was a lichen collection, but I still wasn't very fungal aware. I wasn't into mushrooms or anything like that. It was really it was lichens. And
Shirley Tucker was amazing because she was a plant morphologist, anatomist, but she also she worked on legumes and other plants, but she also was interested in lichens, and that was actually through her father, who was at the University of Minnesota in the plant path department, and so that's where she had got interested in lichens.
Yeah, so same.
Yeah,
same.
So it was a kind of generational transfer there. Yeah, so. You did get interested in fungi later.
I did get interested in fungi later, yeah. So I started university thinking I was going to work on like making pretty flowers and stuff. And then you showed me fungi. I mean, honestly, we had a project looking at labules, and I spent a lot of time looking down a microscope at fungi, tiny little fungi on the outside of termite, and I was just completely mesmerized at that point, like really sucked in a lot of ways. I felt like part of a family as well, right? Like I felt like I was invited into a group of people that were really cool, and the organisms were just had no idea that they were there,
Yeah.
and they're so beautiful. They were just like amazing to look at and sucked in from
Yeah,
there.
and many of the things you were looking at on termites, people didn't know what they actually were at the time, where they
Yeah.
fit into the phylogeny of fungi. And I
Yeah,
should say, I
there were
really saw you less than anybody else, that certainly any of the undergraduates in the lab, because you were so independent. And you worked at night.
I did work at night sometimes, but
Yeah.
also I remember we did termite collecting, which I felt was very very unusual protocol, where you just like go out and like pick up branches and break them open and get all of these termites
Yeah.
out of them. So I spent a lot of time wandering
Lots.
around outside too.
Lots of termites, yeah. So then, after you graduated from LSU with your undergraduate degree
Yeah.
and a couple of papers, research papers with your
Eventually,
name on them,
yeah.
you did what?
I went to Duke University, and I was part of the mycology team at that point, having been in your lab, and I had really good opportunities, and ended up with Rita's vocalist lab, and so I started there pretty like right after LSU, kind of like moved to North Carolina right away, and found, again, on your advice, an amazing study organism that just completely sucked me in from the beginning. So a lot of people, so I worked on Septobasidium, which is an amazing symbiosis between Basidiomycete and scale insects, and was completely driven by John Couch's work. So he was like the guru of Septobasidium, and it seemed
did
like
you meet-
nobody else.
Did you meet John
I
Couch?
did not get to meet John Couch.
Why not?
It was, well, it was a little too late for me.
He had died, what,
Yeah,
20
maybe like-
years before you were there?
I don't think
that
it was
long?
as long. I think it was not as long as that before, but I
I-
honestly don't remember.
Yeah, I had met him when I was a grad student at Texas. He came through there. But I don't remember when it was. But he worked a long time after he was retired, before
Yeah.
he died. Yeah.
Yeah, I
But-
met his daughter,
Yeah.
and hung out at. So he was at UNC and I was at Duke. So part rivalry, part friendship, but very close to each other. And so I spent a lot of time at UNC as well when I was there.
And what did his daughter do for you?
For me, she gave me a book. She gave me his book, which I very much appreciate. And she helped me have access to the Couch collections as well. So she introduced me to the curator at the time at the UNC Herbarium, and we had a very friendly relationship, and I got to spend quite a bit of time looking at his collections and looking through his notes. All of that was held in the library, his library at UNC. So those were available, but it was nice to kind of like hear her take on it.
Yeah.
Yeah.
Yeah, the reason I was interested in Septobasidium was that nobody knew anything about it or had worked on it since Couch.
Yeah.
And he'd worked on other things too that he was known for. He was
it's
a
very
really,
famous for other things, right?
Yeah.
But the Septobasidium just sits there as this complete work.
Right,
not
and it's
revisited.
a book with how many species?
About 180
hundreds? That many?
species in the book.
Yeah.
And then he had a bunch of others that were sort of like ready to be formally described that he didn't quite get done.
Now, is that life cycle that I learned, only a few of the scale insects are actually parasitized? Is that correct?
Yeah. Okay. So can I tell the story?
That's
I love it.
what
Okay.
I want you to tell that, 'cause we need
so
a modern
Septobasidium,
version.
it's the symbiosis, right? So it's a Basidiomycete that infects scale insects, armored scale insects. So armored scale insects have a very reduced kind of life cycle. So from a female perspective, they hatch as eggs, they crawl around for one instar, and then they settle down on a plant and they basically turn into blobs. They have these really specialized feeding mouth parts that go down and mostly hit ray cells, weirdly, so they're not like phloem feeders exactly, so they have specialized feeding, and then they sit there on the plant for the rest of their life. So like where we were in North Carolina, sometimes those females would live maybe 4 months, maybe 2 months, because what they would do is sort of convert themselves into a full biomass of eggs. So they would essentially, their whole bodies they would convert into eggs, and then those eggs would have crawler stages. So this is the normal scale insect life cycle. What Septobasidium does is it has these basidiospores that can infect the crawler stage, that first instar of insect, and then germinates and grows into the insect. And as that insect settles down and feeds, the fungus grows inside of the insect. But it doesn't kill the insect. It actually just uses it as more or less a root to plant food. So the fungus is filling up these individual insects and then eventually completely fills these bodies. Now the insects actually live longer, typically, at least where we were in North Carolina, than the ones that wouldn't be infected. And the fungus fills their bodies and then eventually grows out of their bodies. And so a single infected scale insect gets filled with fungal hyphae, fungal bits, hyphae, haustoria, whatever you want to call them in there, grows out and makes a little tuft. But a collection of these infected insects, those bits that grow out actually form together to make like a mat. And so what they called this for a while were animal lichens, because it looks like a lichen. It's a living branch, so these scale insects have settled onto a living branch, and it looks like, I mean, you might think, if it was a dead piece of dead wood, you might think, oh, this is some basidiomycete that's like eating a bit of dead wood, but it's on a living branch, so it's like a lichen, but in fact, it's only infecting scale insects. And so that underneath that licheny bit would be some of these infected scale insects, but then a bunch of uninfected scale insects as well. So only some of them are getting hit with these basidiospores at the crawler stage. So what you end up with is a mat of fungal hyphae mixed into this lichen-like structure, embedded inside that some infected insects and a bunch of uninfected insects.
Okay.
So that's the story. That's really what most of the Septobasidium species are like.
Okay, so you have this whole covering over all of the scale insects.
Yeah, yeah.
And what does that do for the scale insects?
Oh, good question. Yeah, what does that do for the scale insect? So the main things that it almost certainly does protect from parasitoid wasps. So that's a big source of mortality for adult and young scale insects. So having this mat of hyphae on top of uninfected insects keeps the wasps from being able to find them in the first place and being able to oviposit into them.
Okay.
And then the other major thing is it really changes the water for the main way that young scale insects, when they're crawling around, they're quite susceptible to desiccation. And when they first settle onto a plant, many of them will die just because they get dehydrated. It's environmentally dependent. So maybe it's helping the uninfected ones, but only in the context of parasitoid wasps or sort of like a dry day.
Okay, and to me it looks, these lichens, to me look a little bit like some of the corticioid fungi.
Oh, for sure, yeah.
But they're not related, are they?
No, the Septobasidium is more closely related to rust fungi than to the corticioid, to the sort of like what you might think of as like the mushroomy like basidiomyces.
Yeah, and to me that's just really curious how that might have arisen, how it might have evolved.
Yeah, I guess the idea would be that these ancestrally, the Puccinia mycotina would have been like plant associates, right? And these scale insects are very much in that ecological zone as well, from across the plant, from really roots through to upper shoot.
Yeah, I guess my problem is when I think of rust fungi, I think of crop plant diseases.
Yeah.
But there are other rusts that are rust relatives that might be closer to Septobasidium.
Yeah, maybe.
Yeah.
I don't know.
But Septobasidium never is able to use any of the wood of the
There is one paper that I know of that says Septobasidium has been isolated as like an endophyte from bark from some component. I'm not exactly sure they pinned down where that was from.
Is
But-
that fairly recent, or is it an older paper?
No, it's relatively recent. On the spot, I want to say like two, 2000. I mean, recent. It's like a decade old.
Yeah. Yeah, that's
2010,
recent to
something
me.
like that.
Okay, so
but it's very like that's not where there's that one paper,
yeah,
right?
it's very
That's that one isolated. It's
not the usual, and
Yeah.
it would be a good thing to look at, though, I would think.
Yeah, I think if as people continue to do that kind of like environmental sampling
Yeah.
across plants, you would hope that we can start to recover more about that ecology.
Okay, so let's skip over this. You finished your PhD, and you're looking
Eventually.
for a postdoc.
Yeah. So after my PhD, I got a postdoc with Kathy Aim at the USDA for, it was a pretty short postdoc, and she was working on rust fungi, so that's why I was so excited to go there and work with her. And she had this amazing program where she had collaborators around the world. She was like getting all of these rust collections and just like going through them so systematically, getting this, we were at the time of like expanding our sort of sequence database for everything. So she had this amazing pipeline of just like, okay, we're going to settle what these rust species are. We're going to like fill out the phylogeny of rust fungi. And she let me come in, and she's so tolerant. She let me do other stuff, and I worked on some other components within that rust clade, so other Puccinia mycotina. I was particularly interested in rusts and their ecological interactions, so I started looking at some yeasts that grow as epiphytes on the leaves with rusts, and I also ran into these flies that eat rust spores, these Cichidamide flies that specialize on eating rust spores. So she let me play for a while in the lab, and I had a great time there for about a year and a half, and then ended up in moving on from there to the UK. So Kathy was maybe getting ready to leave the USDA. She had another job that was going to be
And
another
again,
fantastic
LSU
job.
comes into that
Yeah,
because
exactly,
she
back
moved to
to Louisiana,
LSU,
yeah.
where I was,
Yeah, you
and
stole
where
her
you
from.
had been. And
and-
so you went to the UK.
Yeah, I went to work with Matt Fisher in London and completely switched what we were doing. So he was based in a medical school, and I ended up working on Penicillium species. We were in the same location where Alexander Fleming had his accident and found penicillin. And so we were doing a historical kind of project focused on figuring out how lucky he was to find that particular sort of interaction. And we were working in the same location. I had another amazing advisor. It's just like a string of fantastic people making mycology work for me. So he, Matt had us work on that project, but it was a switch because now, although it was kind of about ecology, it was much more medically focused. So we were working a lot on thinking about how fungi interact in negative ways with people instead of insects or plants. At that
Okay,
point.
so a guy who grew up in Louisiana most of his life
Yeah.
goes to England.
Goes to England. I thought it was just going to be for a couple of years, and ended up staying. It was with Matt as a postdoc, and then moved to the University of Bath, and I've been there now for a decade. So settled, completely British. My kid sounds British.
I hear he sounds like an English kid.
Yeah, absolutely. So it was really about people. I just went where people were really nice.
So what do you do in Bath?
In Bath, so Bath is another bit of a switch. I work mostly with industrially kind of relevant fungal interactions. So Bath is very much interested in trying to have
Like fungi are the future.
fungi are the future. Exactly. No, really. And probably the biggest project in the lab is a sustainable alternative for palm oil using yeast oils. So we can kind of like exploit fungal ecology to get something that's a little bit nicer for the planet than cutting down rainforests.
Nice. And
Yeah.
you can grow up big batches of yeast. Huh?
It smells crazy. Yeah.
Does it smell good?
I like it. Yeah, and so sometimes the part of the idea of it is that it's very low impact, so you can just do it outside in like fermenters without any additional controls. You don't have to do like really don't manage anything except just give it a little bit of space to grow, as if you were going to make cider in your backyard or something like that, right? But with this, and the flies, like they're
come.
all over. Yeah, they love the smell of it.
So these yeast, are they lots of different species?
We are mostly focused on some that you guys also isolated from from beetles and other insects. So mostly it's Mechnicovia pulcherrima group, right?
Yeah,
So
yeah.
is it multiple? So we look across many strains. Maybe it's multiple species. I don't know. It's one of these complex.
Yeah, and those are some of the
species
ones
group.
that we were getting from. I think we were getting them from. Well, maybe it was the beetles that ate mushrooms. I think that we were getting them from.
And I think isolated like a species from lacewings
Yeah,
as well. That
that's
was
right, I
very
forgot.
closely related. They're kind of, there's a lot of hybridization and stuff.
Yeah,
in
that was
group.
one of his little summer projects.
Yeah.
Yeah, it's funny how it all fits together like that. Yeah, and so you'll probably stay in England and retire there?
I'll probably stay in England and retire there. I think, 2 years that turns into 25 plus or
Well,
something like that.
And I should say I visited you, and I thought it was pretty nice, and then you were getting a new lab, which I never saw.
Oh, well, come back. You
Yeah,
have to
I'll have to
come
do
back when you...
that. No,
Yeah.
but anyway.
So
and
it is a nice...
it's really nice.
It is, yeah. It's a fantastic place to live, and there's interest. We're not a mycology, like, there's not as big a mycology scene, really, in that part of the UK. But the mycologists that are there are really committed to it, and it feels like it has the whole time, that you've got
Yeah, well, having
friends
more
and
than
family.
one mycologist is pretty neat.
Yeah, true.
So,
Yeah.
because much of my career I was the only one. Yeah.
Well, you had the Plant Path folks, did they not?
Well, one was not real active. Then Kathy
Yeah.
Aim came, and then it was much nicer. But I was many years without another mycologist, really. Yeah.
Yeah, it's
So-
nice to have a team.
Yeah. So anything else that we're missing out of your life story?
My life story, I think I don't think so. Can't overemphasize how much it's been the mentors and mycologists that make it worth doing the mycology. That really is.
Yes.
Every time we're at one of these meetings, it's family and
Yeah, it's fun to-
science. It's like
Yeah.
fun to talk about all of the
Oh, it's solving
mycology.
mysteries all the time.
Yes.
I'm a mystery story reader, and so it's
Yeah.
just logical. I would have liked mycological mysteries. Yeah.
I hear that. Yeah. I
Okay.
think, and
Well, it's great to talk to you.
nice to talk to you
and,
too.
And I know that you were in North Carolina when Reedus retired, or didn't retire, but they had a party for him anyway.
Yeah, well, we had to do it before. I mean, because he won't
to
retire.
make sure that you got it in.
We got to get in there, and then maybe he'll think about retiring now.
Yeah,
that-
Reedus Fest it was called. So
Yeah.
Reedus was a very good mentor to you and many others, and
Yeah.
it showed because people came from all over the place to his Reedus Fest.
It's
a
it's
couple
a
months ago.
community. Yeah.
Yeah.
And I think what is amazing about Reedus is he always managed to make people fit together, just like these places. So we all felt like we were part of the family of mycologists and all part of the same team, but also really excited about the mycology research, and excited for each other's mycology research, and excited to go build boats in his backyard, or excited to go
Play
fishing
music or
yeah,
go
exactly.
fishing and
You know, it was
yeah.
the whole thing, and we definitely felt like a family there.
Nice. Well, thanks for doing this and talking
Thank
to
you
us.
for doing it. I think
and,
this is a great project. I'm glad
well,
that
it's
it's
fun.
it's
Well, it all
happening.
started because of G. Ledyard Stebbins when I heard them do a similar kind of interview with him years ago. It took 20 years to do it, though. So.